in general physics, and then he did his diploma in theoretical physics at ETH Zurich, and went on to theoretical biophysics at Free University of Berlin. He did postdoc work at Leipzig and Harvard before he became a group leader at the Max Planck Institute in Göttingen. Was it the one in Göttingen? Yeah, Göttingen. Right spelling, wrong pronunciation. He was a group leader in biological physics and evolutionary dynamics. And we are very fortunate to recruit him as an assistant professor in 2013. He has won a number of awards, including he was in the inaugural class of Simon's investigators in their new program in mathematical modeling of living systems. So we're very proud of him for winning that, the first round of those awards. Join me in welcoming Professor Oskar Hallatschek.
Can you all hear me? Thanks, Steve, for this very flattering introduction. Thanks to all of you for showing up. This is really the first time since I arrived that I can address the entire physics community here in Berkeley, so it's a particular honor for me, and I hope you came not only because I was announced as ex-chancellor, so I'm really just a small assistant professor that for the first time presents some of the work that I've done here. So the research goal, the general research goal of my lab is to use both experimental and theoretical techniques to better understand collective phenomena in living systems. The basic idea is that when many cells come together and interact, then on large scales, sometimes simplicity emerges that can be understood with simplified effective models that abstract from microscopic details. That general approach, or the hope that this approach works, is deeply rooted in my upbringing as soft matter theorist, where we do this all the time to understand complex equilibrium systems. And I've started to apply this to living systems, and I want to convince you that some of that can work. And living systems are particularly interesting because they are, of course, out of equilibrium. They consume energy in order to proliferate, and thereby they break out of the tight constraints that are imposed on equilibrium systems. In fact, today's talk, I want to focus on proliferation and expansion processes, which, as I show you in a minute, drive many important dynamical processes. I want to convince you that these expansion processes are very, very sensitive to noise, and that has interesting consequences, including that evolutionary success is often a matter of chance than of fitness. But first, let me give you an overview over expansion processes. The most tangible way that I and my students experience those expansion processes in the lab is when we grow microbial cultures. For instance, when we take a test tube and fill it with 5 milliliters of rich medium, drop in a couple of microbes, then they start to divide happily every 20 minutes, and after just half a day, the entire culture is cloudy and filled with 10 to the 9 cells. That, of course, is the miracle of exponential growth that is possible in this extremely well-mixed world. But reality is less well-mixed, and out there in the wild, there are many population expansion processes that are structured in some way, mostly in a spatially explicit way. We can model this, or we can represent this in our lab to some extent by growing microbes on surfaces, which they do in nature very often and form biofilms. Here you see a colony that has grown, and that took much longer to grow, of course, than this well-mixed culture, which grows exponentially. Then even if you look at entire multicellular organisms, we are consisting of many 10 to the 13 cells, and those cells emerge from a single cell through multiple rounds of cell division. This is also an expansion process, an extremely regulated one. In all of these expansion processes, sometimes copy errors occur, which are mutations. Those mutations are essential in driving adaptation, but they can have bad side effects. Microbes can evade antibiotic drug attacks. So that's good from the point of view of microbes, bad from the point of view of humans. And in this case of development, when during this regulated expansion, the wrong mutations occur, you can get abnormal growth and the formation of cancer. This really is evolution, in a way, adaptation. And of course, expansion processes happen on many different scales, not only in Petri dish, but on many scales relevant to Earth, including the entire surface of the Earth. The humans first emerged sometime several 100,000 years in the past in Africa, and then started to spread across the globe and brought not only humanity to distant parts of the world, but also invasive species. This year is a cane toad that was brought to Australia about 100 years ago, and since then is spreading across the face of Australia. It's also adapting, by the way. The cane toads at the front of this expansion have developed longer legs, apparently to run faster. Moreover, there are spreading processes of more elusive items, for instance, of pathogens that travel with humans, or just of information. For me, the most important case is the case of evolution. For evolution to occur, we must have expansion processes. The basic picture here is you have some resident population, which is often called the wild type, and then as the wild type goes through generations, copy errors occur, these mutations, and occasionally a mutation is beneficial. It leads to faster growth of the mutants, and so the mutant emerges and starts to supersede the wild type. And this is also an expansion process, and it controls to some extent how fast evolution can be. So in all of these cases, expansion processes control the dynamics, so it's quite natural to ask, how can we model theoretically those expansion processes? What types of spreading are possible? This will be the question of the first part of my talk. In this first part, I will not talk about mutants at all. That comes, or mutations, that comes in the second part, where I become interested in and what is the impact of these spreading processes on evolutionary dynamics. That in fact many different classes of spread, or several classes of spread exist, becomes evident when we think about epidemic spread. Epidemic spread changed dramatically over time. If you look into history, then in the Middle Ages, we had in the 14th century the Black Death. That was a bacterial infection. The bacteria lived on rats. These rats were brought to the shores of Europe by merchant ships. And from there, the disease spread across Europe, wiping out nearly a third of the entire population. That was the most deadly demographic catastrophe, actually, of modern humans. It took like 3 centuries to recover from this drop. But if there's something good to be said about the plague, is that it swept quite slowly and predictably. Spread like a wave across Europe, and at a speed of around 300 to 600 kilometers per year. So you could have easily outrun it, in principle. That has changed. Nowadays, for instance, here's the example of SARS, which emerged in 2002, somewhere in China, and then it took around 6 months to spread across the globe. This is a very unpredictable, very fast growth, and it frequently occurs nowadays when such worldwide epidemics occur. The big difference here is, of course, not the difference in disease, it's the difference in transport. In the past, when you wanted to move across land, you could either walk or take a horse. Nowadays, we not only have elaborate air transportation network, as is shown here, but we have vehicles really adapted to nearly any scale that's relevant to us. We can walk, we can take the bike, we can take the e-bike, we can take a motorbike, a car, and so on. You see the pattern. That leads to a nearly scale-free transport of humans, as has been observed in several studies now. The first one, which I'm highlighting here, used a very elegant trick to actually track dollar bills as a proxy for human movement. You might have seen dollar bills that carry this stamp, track me at where'sgeorge.com. So this was first a geeks website that wanted to track dollar bills and convince people to go online and type in the serial number of the dollar bills so that dollar bills could be tracked. But what started out as a fun sport then was picked up by scientists that harvested this data set to understand how dollar bills travel across the US. And what they found is summarized here. So P of R is the probability that a dollar bill moves a distance R in a certain time window that you fix. So here the time window is fixed to 4 days. And you see a dollar bill typically moves around 10 kilometers. There's a peak in this distribution, but it has a very broad tail. So it follows a power law over more than 2 decades beyond that. So the question is, can we understand using models the difference in spreading and how the spreading processes depend on the difference in dispersive behavior? The question is of how to model population expansion is truly an old one. Let me give you a brief summary of what has been theorized. The oldest model is due to a geneticist, Ronald Fisher, as well as Kolmogorov, which we know from turbulence physics, who stated that the rate of change of the population density in question, so be it an epidemic or an invasive species, so the rate of change of this population density as a function of position and time is controlled by 2 terms. The first term is diffusion, that just represents random motion of individuals. And the second term is so-called local logistic growth. That term simply works as follows. If the density is very small, as here, for instance, in the tip of this illustrated wavefront, then resources are abundant, and it's assumed that the density grows exponentially. You have a linear term here then, because you can forget about the nonlinearity, and you have exponential growth. But exponential growth has inevitably to stop at some point, and this is modeled here by a nonlinearity, which leads to saturation when the density reaches a limiting value k, which is called carrying capacity. That happens here in the bulk of this wave. And the consequence of this equation is that you get traveling waves where you have growth in the wavefront, which then leads to the filling up of these sites. But during the filling up, you get a rejuvenation of the wavefront, and you have continual growth at the wavefront, which pulls the wave forward. Not very hard to see that the wave speed is given by 2 times the square root of the diffusion constant times growth rate. The scaling just follows from dimensional analysis, and the factor of 2 can be obtained by looking carefully at the linearized equation up there. That was state of the art for 70 years until people took seriously the fact that the spreading processes that one wants to describe usually involve discrete individuals. That implies that there must be number fluctuations, shot noise, essentially. And these number fluctuations could be particularly important because the waves are driven by those few pioneers in the tip of the wavefront where actually growth occurs. Right? And so maybe they thought this is an important perturbation that needs to be taken into account. And so a appropriate noise term was added here. This term ensures that the variance introduced by the noise is proportional to number densities. That makes sense if individuals reproduce nearly independently from one another, like bacteria would. And that leads to a strong correction to the wave speed. So here you see that the wave speed is smaller than the deterministic one, and the correction only very slowly decays to 0 as the carrying capacity goes to infinity. That's a singular perturbation that cannot be obtained by any systematic approach. One can, however, as we have shown, one can actually modify this nonlinearity to obtain exact results. So it turns out this nonlinearity doesn't have to be quadratic. A quartic saturation term would also work. In fact, many other types of nonlinearity lead to the same behavior. So we essentially played around with the nonlinearity and found a way how we can close the hierarchy of moments at any arbitrary level, which allows us to obtain exact results here. that can also be used to understand different kinds of waves. For instance, how microbes adapt and move not in real space, but in fitness space, where we have here as x-axis the growth rates towards higher growth rates. Well, this is a nice model for traveling waves, but it has a serious limitations, especially when we think about epidemics, which are driven by these discrete long-range jumps. Here we assume that we can model movement by diffusion, right? That doesn't work if you have these long-range jumps, so we need to generalize that and replace this by an integral, which sums up all the ways we can jump to position x from other positions x prime. So that gives us an integral. The function g of x minus x prime that appears here is the jump, is the probability density to jump a certain distance, and I will call this jump kernel in the following. Well, for this model, very few results are available, essentially only when we assume that the noise term does not exist. And then the statement is that there are essentially just two behaviors possible. Either this jump kernel is very narrow, then the behavior of this equation is such that we get a wave-like spread, or the kernel is sufficiently broad, then we find exponential growth. So we are back to the way that bacteria grow in the test tube, or essentially on the Petri dish. Well, I want to convince you that this really doesn't hold up to stochastic simulations. And the reason is that these mean field approaches ignore the discreteness of jumps. This term here introduces density everywhere on the x-axis so that you get exponential growth everywhere, and that breaks and is inconsistent with stochastic simulation. The easiest way to do this, to see this, is to do simulations, very simple ones. Imagine you have a two-dimensional lattice, x, y here, and you start with an initial seed at the center. So this could represent, for instance, the start of an outbreak. And then we carry out repeatedly a very simple computational time step. We randomly choose a site from this lattice, and that's our source site. If it's empty, we do nothing and continue with the next time step. If it's filled, then we allow the infection to jump a certain distance, as is shown here. The distance is drawn from a jump distribution that we fix at the beginning of the simulation. Any reasonable jump distribution has a certain short distance peak, which will lead initially to the growth of a little patch. But when this patch becomes larger, the probability of a distant jump increases. And when this occurs, then a second outbreak occurs. And this outbreak then is also growing, the sub-outbreak, until the two outbreaks merge. And the game is essentially to understand the seeding and growth and merging of these sub-outbreaks. The jump distribution that we are using in the following simulation has a tail that is a power law for several reasons. First of all, for the humans, we have seen that the tail is well described by a power law for 2 orders of magnitude, but also it allows us to classify behavior. So I'm assuming, as in this form here, d is the dimension, that is equivalent to saying that the probability that the jump distance is larger than z decays like z to the minus mu. Mu has to be larger than 0 for this distribution to be normalizable. And then we find several interesting behaviors, which I want to illustrate by movie. So let me start with this one. Mu is 3.5. That's the largest, so that's the most narrow jump distribution that I will consider. And I run this movie. You see here in the center, the outbreak starts to grow. It's very noisy, has a very rough boundary. You see long jumps are occurring. But on long time scales, this really just looks like a pancake that's growing at a constant speed. Right? So we can define a radius of this outbreak and measure it in long simulations, and we find that asymptotically this grows at a constant speed. That's already in striking contrast to the mean field results, which would have predicted, well, any power law leads to exponential growth. Any power law in the jump distribution leads to exponential growth. If we lower mu we enter a new regime where the key jumps become longer and longer as time proceeds. And so here's a particularly big one. As a consequence, you get an accelerating wave where the effective radius grows like a power law with an exponent that's larger than 1. So it's accelerating. If mu is crossed even further, there's another phase transition, and we enter a third very rapid regime, almost metastatic growth, where outbreaks appear everywhere, and suddenly the whole field of view is filled. But even this is still slower than exponential growth. It's growing according to a stretched exponential e to a power law where the exponent is smaller than 1. Now, these analytic results that I show you at the center can be obtained by a beautifully simple coarse-graining procedure, which in its essence is purely geometrical. And so I want to convey this idea to you, this geometrical idea, up to the point where the rest is just algebra. Right? But the key idea is really geometrical, and I want to set it up. But by thinking about how an outbreak looks like in a space-time plot. So the x-axis is space, the y-axis is time, and the outbreak starts at the origin. As time proceeds, the outbreak grows, as shown here, and it's an accelerating outbreak, so that this looks like a funnel opening up towards increasing tides. And what I'm indicating here is just supposed to mean the core of the outbreak. So these are the sites that are typically filled by time t, meaning their probability of occupancy is of order 1. That doesn't mean that outside this core there's nothing, but it means that it's very dilute outside this core. Okay? That's important to remember because that means that most of the jumps emerge from the core. For instance, if a jump occurs like that, starts in the core and leads into the core, that is pretty inconsequential because the core is already filled. But if such a jump extends into the dilute region, then that is a seed for a new outbreak. And how will this outbreak grow? Well, at first, it grows pretty undisturbed from the original outbreak. So to illustrate that, I should just copy and paste the original funnel and move it to the new seed as I do in this little animation. And you see there's a second sub-outbreak emerging. Now, I drew this outbreak on purpose such that it contains this gray circle. So I want you to focus on this gray circle now. This is the edge of the original outbreak at time t, meaning it's the most distant site that is typically filled by the time t. And I want to think about ways of filling, of infecting that site. The pathway that I've shown just now is one way of doing that, but it has probability mass 0 because it depends on this particular jump here. This is another way of filling this gray circle, a long jump here. It's contained in the sub-outbreak, or this one here. If you think about all the ways that you can infect this gray circle, you end up with this blue region here, which we call the target funnel. The target funnel has the property that any sub-outbreak that starts there will contain this gray circle. If you think about it a little more, you see that it must have the exact same geometrical shape as the red funnel, the forward funnel, except that it's flipped in time, so that this entire picture is time symmetric. And moreover, you see that because these boundaries are curved the way they are, there's here a nice wide gap appearing between the source funnel and the target funnel, which means that if you want to infect the gray circle, then a jump must exist that goes across this gap. Well, our basic argument is now to say that the expected number of jumps from the source region to the blue region is just about 1, the minimal number of required jumps to actually infect this gray circle. The reason is, if this number would be much smaller than 1, well, then typically these jumps wouldn't occur, and the gray circle couldn't be typically filled, which is what we assume. And reversely, if this number would be much larger than 1, then the gray circle wouldn't typically be at the edge of the core, it would be inside the core. That's the basic argument, and it's an argument, it's a constraint for each time point, and it constrains the dynamics and gives us this phase diagram I alluded to earlier. Let me just give you an idea how this can be mathematized. The expected number of jumps can be calculated by integrating over the jump density, the kernel, from red to blue, over time from 0 to times t. And then I need two space integrals, one for the red funnel and one for the blue funnel here. This is a messy integral equation, hard to solve. But what helps is that on long times, the crucial jumps happen precisely at half time. The reason is that on short times, the source funnel is very small, which means there are just very few jumps emerging at all from the infected region. And on large times, it's the target funnel, which is very small. There are many jumps, but they don't hit the target. And so there must be a maximum in between, and because of the time symmetry, it's exactly at half time. That allows us to carry out a saddle point approximation from this integral equation, and we obtain our main result, which is really this recursion relation, which is a systematic way to coarse grain the system. So we can go from, if we know the size of this core at half time, we can go to the size of the core at finite, at twice that time, given the jump kernel. Now that can be now solved for any kernel that you give me, but the interesting ones are the power law kernels, which show these different phase behaviors. And we can do that and compare the simulations, and this comparison works well. I have a data plot, I can discuss that later on, but I think it doesn't make sense if you stare at this too much. I want to give you more an overview of the phase behavior that we find here is a function of mu. So first of all, as I indicated before, if mu is large enough, then we have just pure wave-like growth. We lower mu, we cross a critical point at d+1, we enter this power law regime, and then there's another critical point, we enter stretched exponential growth. Here are other simulation results where you see the brightest colors are the most early infected sites. This is the asymptotic picture, which is important at finite times. There are these critical points, but these are just points. Importantly, when we go to finite time, it turns out that these critical points, they blow up and become important intermediate asymptotic regimes, which can dominate the entire dynamic, in a region close to that critical point. And so to actually compare with simulation, it's very important to have the crossover forms that describe the behavior from small to large time. In this picture, we can state that clearly the black death swept like a wave. So it must have been in this regime. The kernel might have been broad, but just not broad enough. But modern human travel really falls into this stretched exponential regime. And that's probably not a surprise, because one can show if the epidemic spreads very rapidly, that is actually equivalent to saying that one can travel on that network very rapidly. So what I mean to say here is that we can map this epidemic model to a model of transport of humans on a network, where we ask about the minimal time it takes to cross a certain distance. And it turns out we have the same phase transitions there, and you want your network to have long distance connections such that you are in this very rapidly growing regime. And that's the case for human travel. Turns out to be also the case for long distance connections in the brain between cortical neurons. With that, let me summarize this first part. So the mean field picture that I summarized initially suggested, well, there are two regimes. Either you grow like in a test tube, or you grow like on a Petri dish. But that ignored noise. And when we turned on noise, we found several new interesting regimes which have no deterministic limit. They are completely dominated by noise. We arrived at these results using this coarse-graining method based on these funnels. And there are more regimes to be discovered. As I indicated, that there's an interesting symmetry here, a time symmetry in this funnel picture. In many epidemics that might be broken. For instance, if you can recover from a disease, then you cannot assume that the source funnel is completely filled. There will be a hollow core in the center that will break the time symmetry and lead to different kind of scaling behavior. For me, the most important consequence here is the following. In the introduction, I told you that these spreading processes are important because they control many dynamical processes, including evolution. Now that we've seen that there are so many different regimes, we want to ask, what are the consequences of these regimes for the speed of evolution, for the behavior of evolution? And a big part of my lab tries to do that, tries to understand what happens with evolutionary theory when we pull it away from the mean field centric view that is quite prominent in this field. I want to give you some results on this question, mostly results that I can easily explain given what I introduced so far. So let's again think about an expansion process. So far, we didn't distinguish individuals, right? They were all identical. But during the growth process, copy errors occur. Those are called mutations. And to make them visible, I will use a color here. Imagine we have a growth process and early on a mutation occurs. Then that's a great opportunity for the mutation because as now when the population expands further, then not only these blue wild type cells will expand, but also the mutants will expand and create like a big family of red guys over there. This descendants from that original mutation, I want to call a clone, just to clarify that term. And I also want to assume for now that this mutation is really just like a tracer, like a label that's attached to this individual and all its descendants. So that's the point I just made. Expansion can generate large mutant clones. This is a clone. And that was actually realized very early on in population genetics by Salvador Luria and Max Delbrück, prominent biophysicists, in a really seminal paper in genetics, which also brought them the Nobel Prize at some point. In this paper, they called these events mutational jackpot events because they are extremely large mutant clones in the final population that were initially very hard to explain. But they realized that this is related to population expansion, and the basic idea of their theory is very simple. Imagine the mutation happens in the first cell division. You start with one cell, there's a cell division, and then a mutation occurs. Then you expect that in the final population, 1/2 of the population will be mutant. That's obvious expectation. If the mutation occurs in the third cell, so after the second cell division, then we expect 1/3 of the final population to be mutants. If it occurs in the nth cell, then we expect 1/n of the final population to be mutants, right? So they derived that there should be a power law, statistics for these clone sizes here, and they carried out a very clever experiment to actually measure that. And these broad distributions and jackpot events are also pretty important for biological processes. Here's an example. Imagine you get infected by bacteria. One bacterium enters your cell, your body, and then it starts to grow. At first, you don't notice, right? So the bacteria keep growing until a point where the bacterial population is so large that you develop symptoms. You go to the doctor, get yourself on antibiotics, and try to kill the bacteria. If it happens that such a mutational jackpot event occurred and that these mutants are actually resistant to the antibiotic, then you will just kill the wild type and the mutant cells take over. So you see, you can have bad luck because of these jackpot events. And indeed, this theory developed by Max Delbrück and others about the distribution of these jackpot events is still used today to understand and predict how well drug treatment works and how quickly resistance arises. But of course, this is really just an argument that works in well-mixed populations. And it's not clear that this works in populations that are not well-mixed, like perhaps tumors, where this theory is also applied. And so my postdoc, Diana Fusco, decided she wanted to clarify whether there is a difference in the mutation accumulation process when you grow a population in a well-mixed way or according to a colony. So I'm now describing the experiment that she did. She started essentially with one stock of bacteria, E. coli, and she grew populations in two ways. The first way was completely well mixed in test tubes, exponential growth. And the second way was via colony growth. So individual cells were essentially dropped on petri dishes and then grew colonies. And during this incubation period, of course, mutations arose, and now she wanted to detect those mutations. The way to do this nowadays is to sequence these entire populations. I try to illustrate here what this means. Each of these lines represents one of the bacterial genomes that were sequenced. And the stars mean mutations. So in most of the places of the genome, most of the sites of the genome sequence, you see no difference between the bacterial genomes, but there are sites where you see mutations. And the population sequencing then gives you a frequency count. It tells you that 1 out of these 10 genomes here had a mutation, right? So you get a frequency count of 10%. Here, so as you scan along the genome, you find further sites that are polymorphic. Here, the mutation is present in 30% of the population, here in 20%. And so as she scans along, she gets many of these frequency counts and can plot a histogram. Here, it's a reverse cumulative histogram. The y-axis tells you the number of mutations that you found that were in a population fraction larger than X. And you see the data for the well-mixed curves reasonably well is described by this power law, 1 over X, which is predicted by the Luria-Delbrück theory. The lower frequency end of this curve is essentially determined by how much money we wanted to spend. This was essentially the control case, so we stopped there. All of the data that you see here corresponds to really big mutant clones. So the populations that she sequenced were around 10 to the 9 cells. So a frequency of 0.5% like here is still 10 million cells. Pretty large. So it seems like Luria-Delbrück theory seems to work for the well-mixed case, as we expected. However, when she plotted the data for the colonies, she found a different result. First of all, the curve doesn't seem to obey a particular power law. It's a shoulder-like distribution. But more importantly, it lies way above the well-mixed result. By a factor of 10, you have more of these high-frequency events. Where do all these extra jackpot events come from? That was the question. And to answer this, she wanted to visualize the mutant clones that are rising as a colony is growing. And to do this, we received great help from Ann Dodson and Jasper Ryan here in Berkeley, who developed a very elegant genetic system in budding yeast. So she engineered a budding yeast strain that effectively switches at a rate of once every 1,000 cell division into a mutant cell. And both of these states can be distinguished by the fluorescent color. So there's effectively a fluorescent color switch at that rate occurring. I cannot go into the details of the genetic trick that they used, but effectively it uses a molecular scissor called Cre. That's a protein that can cut out, that is expressed, first of all, extremely rarely in this wild-type strain, but if it is expressed, it cuts out a fragment of the genome which encodes the fluorescent color of the wild type. And the second effect of this cutting is that another fluorescence gene is turned on, and that leads to the color switch. If we then take this wild type, a wild type cell of this strain, and grow a colony, then this is what we see. It's a bit dark, but you see the background is largely blue. That's the wild type. And then you see all these yellow stains here. This is not dirt. These are mutants. Each of these yellow patches are mutant clones that are the descendants of one original mutation, right? And so we have many events here. and ask about the area distribution effectively of these patches. And you see it's very broad. There are small, tiny patches and big ones. Most importantly, the big ones don't occur early, as Luria-Delbrück predicted. They occur rather late. And let's distinguish the two. The big ones, let's call them sectors. They keep growing at the circumference and the bubble, and let's call these other patches that are occluded, let's call them bubbles. Now, Luria's theory doesn't work, but what we've also seen is that all of these clones first arose essentially at the perimeter of the colony, right? So that inspired us to go back to the simulations, very simple simulations that I showed you in the first part, and turn on mutations there to see whether we can arrive at a similar pattern. Indeed, that happens. So this is again in population expansion simulation, but now at a certain rate, mutations are created, and these mutations can be very successful, essentially serve at the circumference of that colony and create these pretty large clones. And again, we see there are sectors and many little bubbles. And in these simulations can also be analyzed at great detail, and we can study the statistics of the mutant areas, the mutant sizes effectively. And this is what the simulation yields. So this reverse cumulative distribution shows two power laws, in fact, that are connected by a crossover here. And moreover, it turns out that if we condition on being a bubble, then we find that the distribution, that all bubbles essentially fall into this early power law regime, the low frequency power law regime, and these sectors fall into the high frequency power law regime. So these two power laws describe the slightly different physics of these two types of clones. And moreover, it's very important, well, we can describe analytically these power laws, and I'll give you an idea of the argument in a minute, but it was really important to have this crossover because that allowed us to actually compare with our data. So we could collapse the data by rescaling this plot with the sequencing data such that the crossover point is identical. The crossover point represents essentially the largest bubbles in the simulation, or the smallest sectors. And upon rescaling, then we could collapse the simulation with the sequencing data. So the sequencing data really falls into the crossover regime, and it's important to have these simulations to actually explain that. But the power law tails that are indicated, they can be understood from an effective coarse-grained model. So here you see how a bubble is created, emerges at the boundary, and then there's a mutant patch which has two domain boundaries, and these domain boundaries carry out a random walk until the bubble essentially is created. We can imagine the genesis of a bubble as follows. When a mutation occurs, then two random walkers are created that live in the perimeter. These random walkers carry out a random walk up until the time where they annihilate. So if we want to understand the area distribution here, we need to understand the distribution of annihilation times of these random walkers. To do that, we have to know the statistics of the random walkers. It turns out they are super diffusive. These lines are super diffusive, as we have measured in the past, and it can be understood from thinking about the roughness of these of the colonies. The roughness of the colonies generates a super diffusive behavior of these domain boundaries. Anyway, with that statistics, then we find on a scaling level that the distribution of these bubbles should follow a power law with this exponent, which agrees with our simulations. We can vary the dimensions. By the way, the other exponent here for the sector is intimately connected to this one. There's an exact scaling relation. At infinite dimensions, we recover the Luria-Delbrück result, which is just a power law 1 over x. That's the mean field result. And in 3 dimensions, we find an exponent that's still pretty different from the Wellmix result. And we think that this one is particularly important to understand the genetic diversity in solid tumors, which are 3-dimensional. Now, so far, I discussed only mutants that are neutral. So there's no growth rate difference between mutants and wild type. What happens now if really selection comes into play and there's a growth rate difference? Well, that can be understood from our effective picture that I introduced. If mutants grow slower, well, then they lag a little bit behind, and there should be a bias for these two random walkers, these two domain boundaries, to annihilate earlier. Right? And that indeed describes what we see in simulations. Sectors do not emerge anymore. And in fact, all mutants are somehow confined to relatively small bubbles. But bubbles still exist, right? Because random walkers by chance can move away from another, even though deterministically you would expect that they annihilate each other immediately. That's the deleterious case. If, however, mutants grow faster, then we would expect random walkers to move away from one another. And deterministically, we would think, well, the mutants take over very quickly. In fact, there's noise. These domain boundaries carry out a random walk. And so most of the beneficial mutations that occur actually trapped here in the bulk of this colony. And only occasionally we get here these discrete establishment events. And I'll show you now quickly as far, how much time do I have left? Oof, not that much, right? Still, I want to show you for both of these cases experimental results and show you that they are in fact relevant. First of all, the deleterious result, the deleterious case is important for drug resistance evolution. It turns out that many resistance mutations are in the absence of antibiotics deleterious, meaning that the mutants grow slower. Right? So then we expect after the incubation period that most of these resistant mutations to be trapped in these tiny little bubbles. And when we then go to the doctor after we have developed symptoms and take antibiotics, we can envision three scenarios. First, if we take a lot of antibiotics, we try to kill as hard as possible as we can, then we kill the wild type, but we liberate the resistant cells, they take over. If we do nothing, that's also bad, because then the wild type just keeps growing. But if we take an intermediate amount of drug, of antibiotics, then perhaps we are in a sweet spot where the wild type is not entirely killed and can still contain the mutants, but overall the growth of the infection is reduced. We can demonstrate this effect to some extent in experiments where we trap a bubble of resistant cells inside wild type cells and then apply different amounts of antibiotics. We do find a sweet spot. It seems to be also the case that something like this strategy could be relevant to cancer evolution. In this paper, one has found that it can be beneficial to apply intermediate amounts of chemotherapy to the cancer. This is, of course, not antibiotics. All right, so now let me go to the beneficial mutations. This is now work by my PhD student, Geometrical. He wanted to study this process of establishment of beneficial mutations. As I told you, in the beneficial case, any case, if a mutation occurs, two random walkers are created that, in the beneficial case, move away from each other. But still, the likelihood of death is pretty high in the early time. So can we see this in experiments? So he did a very simple experiment where he had two types of, two strains of, in this case, yeast. He also looked at different systems. The blue guys are the wild type cells, and the yellow guys grow a little bit faster. And the faster growing strains are mixed with the wild type strain at a very low ratio and then plated on an agar dish. And then we look at the fluorescent images and see a picture like that. In the case of yeast, you really see discrete establishment events of beneficial mutations. So it's, perhaps between 10 and 20 of these events. That's a small number, given that in the inoculum, there were thousands of these beneficial mutants. So most of these initial mutants were pruned by this annihilation process, and only a few of them escaped. Moreover, if he looks at a different system and chooses the same parameters, so the same mixing ratio, the same growth rate difference, he finds that the establishment events can be very different. Here in the case of E. coli, the establishment events he finds are much less. And he could explain it by the difference, it's hard to see here, in the random walk behavior of these domain boundaries. So in the case of yeast, you see here from an experiment where both strains are neutral, there are many, many sectors, and these domain boundaries are nearly straight. So the diffusivity is low, and hence one expects very few annihilation events. Here in E. coli, the domain boundaries are very rugged, and accordingly, one expects more annihilation and fewer establishment events of beneficial mutation. So you see the survival of these beneficial mutants very strongly depends on the diffusivity of these domain boundaries or on the level of noise. And you also see that this level of noise can be large, it can be strongly varying between different systems. And in this case, it has really an interesting biophysical origin. Here you see time-lapse movies of growing colonies of, in the left case, yeast, and in the right case, E. coli. And what's shown here, these colored lines of descent. So you see the ancestry, essentially, of a couple of selected cells. You see that these lines are very straight, very smooth. They are random walks, but the diffusivity is low. In the case of E. coli, the random walks are much, much rougher and much more rugged. The reasons for this difference is a buckling instability that occurs in the system here on the right. E. coli are rod-like. They tend to elongate and divide in the middle, and that has the tendency to form streets. And when these streets become long enough and bump into each other, they essentially buckle, and that leads to this enhanced diffusivity of these domain boundaries on the right. So as you see here, perhaps in these movies, these are very interesting, active, granular systems. And really just as a teaser, in the end, want to highlight actually what a big other part of my group does, tries to understand essentially the soft matter physics of microbes viewed as an active granular system. So here we took yeast populations and grew them in very confined environments with a microfluidic chamber. And so each yeast cell here has a diameter of 5 microns that gives you the length scale of the system. And in principle, so all the motion, by the way, all the motion that you see here is pushing motion due to the growth of the cell. So the cells are growing here in this chamber, and they should all flow out through this channel because that's the way they came. But it turns out they behave a little bit like sand flowing out of a funnel. They jam up at this outlet, and because of force chains that are forming across this outlet, as a consequence, they generate a pressure inside this chamber, which is still increasing, as you see here, 8 bars or 0.8 megapascals. It's an extremely high pressure, much larger than in your car tire, and that feeds back on the growth of these cells. And as we've shown, it can also destroy brittle materials. So we suspect this phenomenon to be important when biofilms grow in porous materials, and might explain might explain biofouling there, a mechanical mechanism. But I think these systems are also interesting from a physics point of view because there is local injection not only of energy, but of degrees of freedom due to the division of cells when cells divide. Cell division here leads to a local melting of any rigid structure that forms, and that leads to interesting physics. At any rate, I think all of these examples have shown that when you have an expansion process, it's often driven extremely sensitive to noise. The reason is that expansion processes are driven often by the few individuals that are most advanced. And those individuals, by definition almost, are small in number, and therefore fluctuations are important. And these fluctuations have important consequences, which are summarized here. I discussed different universality classes of spreading, and I discussed how this can influence the genetic diversity of populations. We have an excess of jackpot events, and that this can influence the course of drug resistance evolution, and that generally hampers selection for the fittest. Let me thank my group for being so much fun to work with and also for working so hard. Diana Fusco and Matti Gralka were really important for the work that I presented here. Diana Fusco worked on the generalization of the Luria-Delbrück experiment. Matti Gralka worked on the establishment of beneficial mutations in colonies. And lastly, I just flashed work of nearly 1/3 of the group, which works on trying to understand the soft matter properties of these active granular systems. So Morgan Delarue is the main experimentalist here. I want to thank in Berkeley, Jasper Ryan and Ann Dodson for their help with this genetic switcher strain, and Daniel Fisher, with whom I worked on this generalization of this generalized model of spreading processes. All right, with that, I take questions. Sorry for going over time, I guess. In microbial systems. So this is driven by the large osmotic pressure difference between inside those microbes and outside. And that can only exist in cells that have a cell wall. Otherwise, they would explode. Normally, you don't see this pressure because it rarely leads to a pressure on a larger scale. But when they jam, as in this system, that occurs. Well, so we have to generalize the basic model to 3-dimensional, of course. The precise quantitative results would be different, but in 3 dimensions, as we've also shown by 3-dimensional simulations, you also get the structure of 2 types of clones. One is bubbles, the other one are sectors. And when the mutations have a disadvantage, then most of them are really trapped in little bubbles inside such a sphere. And is that relevant? Well, we think it's relevant in systems with spatial structure. Where you have growth largely in some growth layer at the perimeter. And many solid tumors are of that type. So we think it could be important there. We haven't shown this in any way. Well, it turns out there's some helicity associated with these domain boundaries in this system of E. coli. And that also seems to have a microscopic origin. There is, during the growth of the cell wall of these E. coli, there is a sewing process going on, actually, kind of a molecular sewing process, which has a particular helicity. And that, in collaboration with Casey Wang in Stanford, we're currently investigating also by simulation. It seems to be the case that that can explain this helicity, but it's definitely there. Right. Yeah, in general, I think okay, so the question was whether, well, I introduced, when I showed you these time-lapse movies of growing yeast cells, which are spherical, and E. coli cells, which are rod-like, I highlighted that there are buckling instabilities in the rod-like case. And what's important there is to understand what's the characteristic length scale at which you get buckling. First of all, the length scale is much larger than a single cell, which already indicates that this is really probably a physical process, because how could the cell know about this length scale if it's much larger than itself? Secondly, this length scale is, of course, dependent on the parameters of the system. So I suspect, we haven't done this, but I suspect if you change the physical properties of the substrate, you can change this characteristic buckling length scale. But given you know that length scale and this diffusivity of these domain boundaries, one can predict to some extent what will be the outcome, what's the probability of establishment of these beneficial mutations. So the basic idea is you have these two random walkers. Diffusivity tells you how strong, how likely they annihilate quickly, and then it fights against this bias. Right? And the competition between the two tells you, gives you a probability of actually surviving. Well, what would I expect? I would also expect that based on this coarse-grained picture, where we have boundaries between the two types, that I think will still pertain. But if like one type eats up the other or converts the neighboring type into its own type, that will lead to an extra bias and acts like a selective advantage to some extent. But there are lots of things that haven't been worked out yet, like frequency-dependent, so-called frequency-dependent selection, where there is, so what I've shown so far was really just very constitutive effects, where mutants are either always growing slower or always growing faster. But what happens if this growing faster or slower depends on the proximity to the other type? That's so-called frequency-dependent selection. It leads to interesting effects which haven't been worked out in this